All right, so this week we're going over enzymes. So we'll talk a little bit how enzymes work and how they can help life persist. Okay, so by the end of this module, you should be able to do these things. You should be able to understand how enzymes and substrates are specific for one another, how induced fit works and how that increases the rate of chemical reaction. You should understand these terms, substrate, enzymes, and active sites. You should understand a little bit of exactly what's going on with how these enzymes are catalyzing reactions. And then you should also be familiar with the factors that affect enzymatic activity, specifically how substrate concentration, pH, and temperature can affect these enzymes. And fundamentally, as we go through this, you should understand why enzymes are so important. To understand enzymes, let's talk about an example that we already know where an enzyme is used, and that is with hydrolysis during digestion. So we talked about this a little bit with the macromolecule lab. So if you remember, hydrolysis is when we intake our food and then we break down those macromolecules into their monomers in the presence of water. So we can't use these monomers until we break them down into those monomers. And we do this using hydrolysis. However, hydrolysis takes a long time, so usually you're going to need an enzyme to help speed up this reaction. So this hydrolysis reaction is actually really slow, and it's too slow for life to exist relying on it alone. So that's why we have these enzymes to help speed up these reactions, and that's exactly what enzymes do. They don't cause chemical reactions, they just speed up the chemical reactions that could occur. In a lot of instances, you are only going to get these chemical reactions in the presence of high temperature, and usually in such high temperatures where cells can't survive. So that's why we need these enzymes. So we don't rely on temperature or just rely on how long it takes these reactions to occur. We can use these enzymes to speed things up in a reasonable environment. Okay, so when we're talking about enzymes, we're talking about proteins, but it's important to understand that proteins aren't only enzymes. Proteins can do a lot of different things for the cell and a lot of different things for your body. Just remember too what proteins are made out of. So their monomer is amino acids. So proteins are these long chains of amino acids. And based on the sequence of the amino acids, going to give proteins different shapes and different functions. So proteins can be used for things like structure or regulation or transport, but primarily what we're going to talk about today is their function as enzymes. All right, so an enzyme is something that catalyzes a reaction, and by catalyzing, we mean speeds up the reaction. So just like we talked about with the monomers of food, how we broke it down using hydrolysis. Well, water is actually causing the reaction there, but it's really slow. So there's enzymes that allow that reaction to happen faster. And this is what is going on to increase the reactivity rate. So enzymes are able to catalyze these reactions by reducing the activation energy. All right, so let's look at these graphs to kind of understand what activation energy is. So during a chemical reaction, we always have reactants and a product. So the things that are there before the reaction occurs and what is made after a reaction occurs. And usually these reactions go down in their energy state. So molecules naturally want to be at a lower energy state. So let's say here on this chart, we have some sort of reaction. Let's say this reaction is two different molecules that want to come together. Separately, these molecules might have a higher energy state, but after the reaction occurs, they're going to bind to a new molecule that would be in a lower energy state. So they naturally want to be here. However, sometimes it takes energy to save energy, and you might need to add energy to these reactants to get them over this hump before they will naturally go to this lower energy state. And we call this hump the activation energy, the amount of energy it needs or the amount of energy required to push this reaction before it will naturally go to a lower energy state. So sometimes you need to add energy for this to occur. And this is where enzymes come in because enzymes will actually lower this activation energy. So when you add an enzyme, you're actually lowering the activation energy, making it easier for these reactions to occur. So it requires less energy in this figure to push these molecules to a point where they will naturally go to a lower energy state. And another way we can think about this is the frequency of the reaction occurring as well. If we go back to this chart on the left here, if you have a high activation energy for this reaction to occur and you have tons of these reactants, you're probably not going to have too many that actually make it over this energy state. Versus if you add an enzyme, back to this chart over here, even if we have tons of these reactants and the potential for this reaction to occur, these reactions are going to happen more often because of this lower energy state. All right, so let's talk a little bit more about what enzymes are and how they are formed and what ultimately gives them the ability to catalyze these reactions. Now, again, when we're talking about enzymes, we're talking about proteins. And with proteins, we have these sequences of amino acids, and different proteins will have a different sequence of amino acid. And based on these different sequences, you're going to have different structures and functions for these proteins. And you can think of all the different combinations you could have of amino acids. All right, it's almost infinite. And this ultimately results in millions of different types of proteins. All right, so let's go through this figure real quick to see what's going on here. So we have these chains of amino acids, and each amino acid has its own unique chemical characteristics. And based on these chains, they will cause different folding patterns of this chain. So sometimes you might have certain types of amino acids that come together that will cause the chain to coil around each other. So these amino acids are reacting to each other to cause this coiling effect. So we have a term for this conformation. We call it an alpha helix. While sometimes you're going to have amino acids that are in a certain chain that are going to interact with each other to cause more of a sheet-like conformation, and we call these beta sheets. And so you have these proteins that are making up these alpha helixes and beta sheets. And we commonly refer to these as secondary structures. So once you have formed these alpha helixes and beta sheets, these are then going to interact with each other as well to cause the next folding or next structure of this protein, which we call the tertiary structure. So these beta sheets and alpha helixes are going to fold around each other to cause these three-dimensional structures here. So these tertiary structures for a lot of proteins is the last stage. Once they fold here, they're able to do or achieve whatever function that they have. And their function is very specific to how this is folded. So what amino acids are placed where in this protein and how this folding is going to occur will allow that protein to achieve its function. Sometimes the functional structure could be this quaternary structure where you have multiple proteins that come together to create a protein complex. So a good example of this is hemoglobin. Hemoglobin is a protein complex of multiple proteins that come together to cause some sort of reaction. Okay, so just to recap this, we have a primary structure, which is your amino acid chain. Based on the properties of these amino acids and the order that they're in, they're going to cause either an alpha helix or a beta sheet for your secondary structure. And then these alpha helixes and beta sheets are then going to interact with each other to cause a tertiary structure, which in a lot of cases, this will be the functional protein. And then sometimes you could have multiple tertiary structures come together to form a quaternary structure, which is a protein complex that will then achieve whatever function the body needs. Okay, so what are we doing here by making these three-dimensional structures? Well, somewhere on this structure, there is going to be amino acids and chemical characteristics that's going to accept some sort of molecule. All right, and these are going to be very specific to the molecule that they interact with. So the term that we use for these molecules that are going to interact with these enzymes is substrate. So a substrate is a molecule that's going to come in and interact with this enzyme. And where that molecule attaches to this enzyme, we call it the active site. So the active site is where these molecules are going to actively bind to the protein. Just looking at how complex this system is of making these proteins, you could see how specific these proteins can get to the substrate that they attach to. So these are very complex structures that are forming in such a way that it can only bind to a very specific substrate. And so we have a term for that. We call it enzyme substrate specificity because these enzymes are so specific to the certain substrate that they interact with. Okay, so what is going on after these substrate attach to the enzyme at the active site? Well, the current theory is the theory of induced fit. An induced fit is the idea that this active site, the part of the enzyme that the substrate attaches to, will change its shape after the substrate attaches. So if the enzyme function is to bring two molecules and have them bind together, you might have two molecules that bind on two different active sites, but then that protein is going to change its conformation to kind of force those two molecules closer together to make their chemical reaction more likely to happen. Or if the function of the enzyme is to pull a molecule apart, well, you're going to have the molecule fit into the active site, and then the enzyme is going to change its conformation, change its shape at the active site to make it easier for that molecule to be pulled apart or for the chemical reaction to split that molecule. And so this induced fit is actually lowering the activation energy of these chemical reactions in some way. Okay, so let's look at a more realistic 3D animation of an enzyme and the molecules that interact with it. So this is just a video I found on YouTube. I mean, it's great to see all these pictures that we have in the PowerPoint, but sometimes it helps to actually visualize these things if we get more detailed with the illustrations. So here we have this enzyme that's in blue. So these are all amino acids that I have folded into a specific protein. And here's our active sites for the two molecules that are coming in. And notice once the molecule enters this protein, the protein changes its shape. This is the induced fit that we're talking about. And because this induced fit causes the chemical reaction to occur between these two molecules, and then the product is released. So these foldings of these proteins can be very complex, and it ultimately results in a very specific active site or sites on this protein, which kind of limits how many different molecules this protein can interact with. So typically, these enzymes can only interact with a very specific molecule or molecules, and hence influence a very specific chemical reaction. Okay, one very important property of enzymes is that they're not used up in these reactions, meaning that they can do the same thing over and over and over again, and you don't need to make new proteins to have these reactions occur over and over and over again. So that's what this diagram is indicating. So here we have our enzyme. It's going to pick up the substrate at its active site. After that attaches, you're going to have induced fit occur. So this enzyme is going to change its shape to cause the chemical reaction in this substrate. After the chemical reaction is done, the product of the substrate is going to be released, and then that enzyme is free to do the whole thing over again. There is no need to make a whole new protein. This protein can do the same thing over and over again. And this is an important point for life to exist, because if we had to make these proteins over and over again, it wouldn't be efficient enough to allow a cell to stay alive. So we need these enzymes to be able to be reused in these reactions. All right, so now we're going to talk a little bit about the different conditions that can affect the ability of an enzyme to function. So primarily, we're going to talk about temperature, acidity, how much substrate is available, and inhibitors. All right, so first let's talk about temperature and how temperature can affect an enzyme. Okay, so let's take a look at this chart here, and let's say we just put a bunch of enzymes in some sort of beaker and add some substrate and then watch the chemical reaction occur. And let's say we did this a bunch of times at a bunch of different temperatures. And then we measured how fast these chemical reactions were occurring, how fast these substrates were turned into their products. And we'd most likely see a chart like this. All right, at lower temperatures, we see less enzymatic activity. The enzymes are not being as efficient. And at high temperatures, we see less enzymatic activity. Again, these enzymes are not being efficient. And at some point, there is going to be an optimal temperature. There is going to be a temperature that works the best for those enzymes. All right, but why is this happening? What is happening on these two tails to cause less enzymatic reactions? Well, let's start with the lower end first, at lower temperatures. When you cool a reaction, it slows down how much energy is in the system. So at cooler temperatures, since there's not as much energy being released by these molecules, they're not bouncing around as much. And so if molecules aren't bouncing around as much, the likelihood of them reaching their active site of an enzyme decreases too. So imagine a bunch of molecules bouncing around, and whenever they hit the active site, then that enzyme will cause a chemical reaction. But the slower these are bouncing around, the less often they actually make it to these active sites, hence slowing down this reaction here at colder temperatures. All right, so that's what's going on at colder temperatures, but what's happening at higher temperatures? Because theoretically, we're increasing the temperature, these molecules should be bouncing around more, and you would think that they would reach the active site more, meaning that the chemical reaction would happen more. But at higher temperatures, what's being affected is actually the enzyme itself. So those amino acids that are in a very specific sequence and in a very specific conformation of the shape need a certain temperature to have that shape. When you increase the temperature too much, it causes these proteins to unfold. So these amino acids are not interacting with each other the way they're supposed to have the correct shape of the enzyme, because you have too much energy in it. And so these higher temperatures causes these proteins to unfold. And when the protein unfolds, it loses its ability to achieve whatever function it's supposed to do. And so we have a term for this. We call this denaturing. So these proteins are denaturing or are unfolding because of these higher temperatures. And as more and more proteins are denaturing, there is less active sites available because the proteins aren't functioning the way they should. And we have a decrease in how much reaction can occur. Okay, so that's what's happening at lower temperatures and higher temperatures. There's also an important thing to point out here is that not every enzyme is the same. Not every protein is the same. Some proteins have a different optimal temperature than other ones. If you think about proteins that are in humans or enzymes that are in humans, well, a lot of times you're going to see that their optimal temperature is probably what the body temperature is, around 98.6 degrees Fahrenheit. But if you look at enzymes of, let's say, a fungus that releases enzymes into the environment to break down food, well, you're probably going to see a different optimal temperature for those enzymes. So it's important to keep in mind where these enzymes are found and how they're being used and by what organism, because you're going to have different optimal temperatures for those enzymes. All right, next we're going to look at pH and understand what's going on here. All right, so similar to temperature, enzymes also need a certain pH level to take their correct conformation, to take their correct shape. Now, when we're talking about two pHs, we're talking about two different sides of the scale. So we could have a base or more alkaline side, or we could have a more acidic side. So bases are usually between 7 to 14 pH, while acids are usually between 0 to 7 pH. And again, these enzymes need a specific acidity in order to have their correct conformation, in order to have their correct shape. So let's take a look at this chart here, and we again see enzymatic reactions of these enzymes. At lower pHs, at more acidic pHs, we're going to see that the enzymes are losing their shape too much to actually achieve their function. And as we get closer to their optimal pH, we see that more and more enzymes have the correct shape, meaning more and more reactions are occurring. And then we'll see the same thing happen on the other end, where we get into more basic solutions. We're going to see a reduction in the amount of enzymes that have the correct conformation, and hence we're going to see a reduction in enzymatic activity. So again, we have this optimal pH for certain enzymes, and just like we saw in temperature, different enzymes have different optimal pHs. Think about enzymes that are in your blood versus enzymes that are found in your stomach. Do you think they're going to have the same optimal pH? Well, most likely not. Which one of those two things do you think is going to be more efficient in a more acidic solution? Where most likely the enzymes found in your stomach are going to be more efficient at lower pHs, at more acidic solutions than the enzymes in your blood. So your enzymes in your blood might be around 7 pH, while the enzymes in your stomach might be more efficient at a 2.5 pH. And we actually use these characteristics of enzymes in produce a lot, specifically trying to preserve food. So whenever you see an apple brown, that's actually because of an enzyme in an apple that is being released. So one of the things they could do to keep the apple lasting longer is to add an acidic solution to the apple. So a lot of times if you buy apple slices in a bag or something, they add ascorbic acid to the apple because that acidity that they're adding will change the shape of the enzymes that are in the apple that would cause browning. So you're kind of slowing down that browning by lowering the pH of the apples. That's also one of the reasons that canned tomatoes or anything that is tomato based has such a long shelf life, because tomatoes are actually really acidic, and it changes the enzyme shape of a lot of the bacteria or other organisms that would decompose this food. All right, another condition we need to consider is substrate, or how much substrate, or the concentration of substrate that is in these systems of these chemical reactions. So typically, if we kept the concentration of enzymes constant, but we kept increasing the substrates concentration, then we would see this slope of enzymatic reaction. So let's see what's going on with this slope. At the lower end of this slope, we have enzymes, but we don't have that much substrate. So the likelihood of an substrate making it to the active site of an enzyme is pretty low. But as we increase the amount of substrate that's in the system, the higher the likelihood enzymes are going to attach to a substrate or the higher the likelihood a substrate is going to bounce into an active site of an enzyme, and you're going to see an increase of enzymatic reaction. But at some point, you're going to see this plateau here, all right, which is different than the other conditions that we talked about. So what's happening at this plateau is that you have so much substrate that the enzymes are always going to be attached to a substrate. So once a substrate enters an enzyme and the chemical reaction occurs and the product is released, you have another substrate right there ready to take its place. We see this plateau of reaction rate, and that's because all the enzymes are saturated, meaning all the enzymes are being used simultaneously. So it doesn't matter if we keep increasing the concentration of substrate here, because you're going to be limited by how many enzymes are actually present. All right, so that's what this GIF is trying to illustrate as well. This is probably not relevant for most people nowadays. This is a scene from I Love Lucy, which was an old TV show. If you want to go look it up on YouTube, you can. And here, really what's happening is that there's too many chocolates coming down, while these two workers are the only workers that can actually potentially do anything with these chocolates that are coming down this conveyor belt. So these workers are the limiting step. So if you go and watch this video, just pretend that the two workers are the enzymes, while all these chocolates are the substrates coming down and trying to interact with these enzymes. It doesn't matter if you keep increasing the amount of chocolates that are coming down. These workers are already working as fast as they can, so productivity is not going to go up. So the last thing we're to look at is inhibition and how that can affect an enzyme's reactivity rate. So sometimes these enzymes can have an inhibitor or a different type of molecule that will prevent the enzyme from functioning properly. So sometimes these cells don't want a chemical reaction to occur if there is a certain molecule that's already present in the system. And we call these molecules inhibitors. And there's two types of inhibitors. We can have competitive or non-competitive inhibitors. So looking at competitive inhibitors, a competitive inhibitor is an inhibitor that fits into the active site of the enzyme. And when it binds to the active site of the enzyme, it prevents the substrate from being able to bind to that enzyme and prevents this reaction from occurring. So if this inhibitor is present, this substrate will not react. So if this inhibitor is present, the substrate can't bind to this enzyme and the reaction won't occur. Now comparing that to non-competitive inhibition, this happens when an inhibitor binds to a different part of the enzyme. So it won't bind to the active site, but it has its own space to bind to. And how this can prevent the enzyme from functioning is when this inhibitor binds to the enzyme, it could change the conformation of this enzyme. These new chemicals, so this new molecule that is added is changing the chemical composition of this enzyme enough where it will change its shape and kind of ruin the shape of the activation site or active site, which is going to prevent the substrate from actually attaching to that enzyme. Okay, so that's the last thing we're going to talk about for enzymes. So hopefully you have a better understanding of what enzymes are and how they work.